Welcome, brothers. My name is Jonathan Chaffin. I'm the Director of Member Engagement for the Psi Upsilon Foundation. Thank you for joining us tonight for our virtual speaker series. Our speaker tonight is Adam Boyko, Omicron 99. Adam will be speaking to us tonight on his dual roles as a geneticist and an entrepreneur. So he wears multiple hats. Some brief housekeeping notes before we get into the presentation. The speaker series and all of our programs, publications, outreach, and scholarships are funded by your generous contributions. As always, thank you for nourishing our brotherhood in this way. About the presentation, type any questions you would like asked using the Q&A box. If you're not familiar with Zoom, by now we probably all are. If you're not, look at the bottom of your screen and you'll see the Q&A box. We aim for a moderated conversational tone. What that means is I'll try and work them in as Adam and I are talking, or we'll get to them explicitly towards the end. If just to hit a few high points of Adam's CV real quick. He has traveled the world investigating dogs and their genetic origins. He is an associate professor in biomedical sciences at Cornell University College of Veterinary Medicine. Started at Urbana-Champaign in ecology, ethology, and evolution, and computer science. Got a master's in computer science and a PhD in biology.
Thanks, Jonathan. Yeah, it's really exciting to be here. I don't know, do you want me to start with just a little bit of background, how I wound up in Ithaca, or?
By all means.
Okay. So yeah, I as all the Omicrons on here, I started off kind of studying monkeys and computer science while I was an undergrad, and sort of made the decision that I wanted to be a biologist that knew how to use computers rather than be a computer scientist that kind of knew a little biology. So I went to get a PhD in biology at Purdue, because that was kind of the closest good school, and Kelly was still 2 years behind me at Illinois, so it was made the long distance relationship work that way. I was studying butterflies then at Purdue. And, by the end of it, getting a master's in computer science and having a biology PhD, I pretty quickly realized that the field I wanted to be in was genetics. I mean, that's what you do with those two things. And so I was fortunate enough to get a postdoc at Cornell in a lab which was studying human genetics, which was kind of where the big data sets were at the time, and working on some human projects, and then sort of lucked into the fact that there was a dog project that started in the lab collaboratively with other labs, and I started working on that project. And of course, it was looking at purebred dogs, which are interesting for a whole bunch of different reasons. But me coming from this field work kind of background is like, well, there's dogs everywhere in the world, and they're not really purebred dogs for the most part, and nobody's studying them. And so I got some seed money, eventually got some Obama stimulus money, eventually got National Geographic money, and so kind of turned that into a project of just studying all kinds of dogs, and took us to some cool places, got to do some cool research, meet some nice people. Hey, John. And and so my postdoc moved to Stanford. I was there for a couple years, and then Cornell had a faculty position in biomedical sciences, specifically for canine genomics. And so I applied to that and wound up back here in Ithaca doing that. And I thought I was just going to be an academic for the rest of my life. But then it turned out to do the research I wanted to do. You couldn't really get government funding to do big enough studies. And so the idea of, well, if we had a DNA test where people were paying to do the genetic analysis, suddenly we could do a lot cooler kinds of research. And that was sort of the idea behind, doing a commercial DNA test and starting Embark.
So your adventures mapping canine genetics have taken you all over the world.
Yeah. So we started out by happenstance. My brother was going to Africa anyway, so he was doing public health work, and so we did a study, got published in PNAS with dog samples that he collected there. And then we expanded that out to do more of a worldwide, sort of collection. And so I've traveled with him to many places, and we've had collaborators in other places. And we've been to some really nice places and other places where we probably wouldn't have been if it wasn't for this, which is also interesting. It's always fun to visit a place and not kind of be like the typical tourist, but kind of be there for a reason that kind of gets you elbow to elbow with, locals and collaborators and really getting to see how they live day to day.
Sure. Now I read that some of your work involved pariah village dogs.
Yeah. So it was, starting out, a lot of people were trying to dissuade us from doing that. It was like, well, these are just going to be mutts. And I'm like, well, maybe people have brought like German Shepherds and Poodles all over the world and they sort of crossed and that's what they are. But I think for the most part, these are actually like populations that have been around before we were doing pedigree dog breeding. So they're not like mixed breed dogs. They're like actually natural populations, right? Like there's 50 different breeds of rabbit, but like the rabbit you see in your front yard is not a mixed breed rabbit. It's like a natural rabbit population. So, and so we, doing the initial sampling in Africa, it was really clear. Yeah, these are not mixed breed dogs at all. They have very unique genetic signatures. And so that kind of gave us the idea that this is a really interesting population for understanding where dogs originated from, because it has been a contentious sort of scientific debate. And the data weren't very good for addressing it because everybody was studying purebred dogs. And so if you want to figure out where dogs came from, it would be like trying to figure out where humans came from just studying European royal families. Like you're not going to get the actual genetic signatures that you need to solve that problem. And so we sampled around a bunch of places. One of the postdocs in my lab went up to Nepal. We had a collaborator that went to Mongolia. Ryan went to Vietnam. I went down to Peru. Like we sampled all over. And it was really clear that you had this higher level of genetic diversity, like you would see at a species origin. Like in humans, you have the highest level of genetic diversity in East Africa, because that's like the cradle of human civilization. And in dogs, it turns out it's in Central Asia, which is not a place that people had. Some people thought it was in Europe. Some people thought it was in East Asia. Some people thought it was in the Middle East. And if you look at different patterns, you can see why they thought that, but you can also see why that's not the right answer. And so it's pretty clear that dogs originated in Central Asia and then diversified out from there.
That's very cool.
Yeah. Nepal's a really neat place. It was fun because I went back there for my brother's wedding. So he met a girl at Yale from Nepal. And so there was a traditional Nepalese wedding. And then there, we of course sampled a few more dogs while we were there. But this was after the research paper had come out. And so as we're there sampling dogs, people walk up to us and they're like, "Oh, did you know dogs came from Nepal?" I went, "Yeah, we're aware of the research." Yeah. So I guess it turned into a point of play.
That's really cool. So I don't know exactly how many aspiring or practicing biologists or geneticists we have in the audience, but do you have any observations about field work versus lab work that might be of interest, or stories about that?
I mean, field work is fun because you're really problem solving. Really, like no 2 days are the same. You've got to figure out government bureaucracies and travel hassles and weather and improvise solutions with limited resources and all of that kind of stuff. For trying to build an academic lab that's focused on kind of big data science, fieldwork takes a lot of time and attention, and it's not the most efficient way to get data all the time. But then again, if you're the one who has unique samples, then that, gets you a lot of collaboration. So it's this sort of trade-off. Like you don't want to spend all your time in the field and not analyzing and writing grants and mentoring, students and teaching and all that kind of stuff. But if you don't do any of it, then you can sort of be seen as a data parasite. And so, yeah, there's different ways of thinking about it.
Sure.
But I think it's good background, especially when you're a student and like you don't have obligations, like you don't have a family, you don't have grants, classes you need to teach. Like definitely go out and do it, because it's going to give you this big respect for, oh, like I'm reading this scientific paper. Why did they do it this way? And it's like, oh, well, it's because like it's a huge, like you could say in theory it would be great to do a double blind, and have 10,000 samples. But then when you look at the realities of how fieldwork works, you're like, yeah, that experiment would be like impossible to do without like $50 million and 20 years to do it.
Gotcha. Now, you mentioned teaching. You are an associate professor.
Yeah, I got tenure. I just got back from sabbatical, so it's interesting time to come back from sabbatical.
What were you working on?
Yeah, it's sort of a research vacation, and so I spent it with Embark, so
okay,
got a chance to work full time there, for a year with minimal university obligations, which was kind of nice. And we've been able to, reorganize the science team. We've been able to scale up the company. We've got an offer out now for a research director position that should be able to take care of the day-to-day scientific management of some of the stuff going on. So it was really a wonderful opportunity to be able to do that and then travel back and forth between Ithaca and Boston, doing that for a while.
Well, since let's transition to Embark a little bit. You speak to how you started the company.
So from day one at Cornell, when I started the faculty position, I was kind of proposing that, and I don't know how familiar people are with 23andMe, the dog genetic testing company. But I was like, if we had something like this in dogs, we would be able to do a lot better research than what we can do now. Like dogs are great for doing research. They've hundreds of different genetic mutations have been discovered in dogs and studied. But a lot of the promise of dogs is that, we've bred them to do different roles and for some, extreme, looks and behaviors and stuff. Mm-hmm. So we could really use them as a way to understand what are the genetics of behavior. What are the genetics of cancer? Like cancer risk is very different in different breeds of dogs, and that's because of genetics. What are the genetics of aging, right? Different dogs age at very different rates. Like there's a twofold difference in how fast a Great Dane ages versus, how fast a Chihuahua ages. And a lot of that is size, but is it certain size genes that are causing it or other ones, or is there stuff besides that causes it? So there's all this potential for dogs to study kind of complicated biological and genetic problems, but there's no real funding source where you can study hundreds of thousands of dogs, which is what you need in order to answer complicated genetic questions. And we know that from human genetics. So there are now human genetic studies studying diabetes that have over 1 million participants, or heart disease, or and so the only way we, like NIH mainly funds human genetic research. So you're not going to get $100 million that you need to do that study. But if you can get a company that can do dog DNA testing, then you can get, if you do the test right, you can get research data from that. And if you maintain contact with the customer and you're able to get veterinary information and other kinds of information, suddenly you can turn that into a research thing. And so it's very much like a 23andMe model. And so, I proposed that at the vet school. We actually had a company that gave us a $500,000 kind of seed grant to kind of get started on that project. Mm-hmm. And it was successful. We got a number of publications out of it. We kind of built a reference panel that could be used for doing dog DNA testing with like Ancestry and things like that. Mm-hmm. And then when it came time to negotiate a license, the company decided that it wasn't really in their wheelhouse. They do, they're primarily a pharmaceutical company. The genetic testing wasn't really a druggable target, all that kind of stuff. And so the IP reverted to Cornell, and, kept pitching the idea to other companies in the space where I thought maybe they would be interested. And it just was a little bit too out there. It wasn't quite anybody's vision for their company, like how to get this product to work, even though like in my head, it made a ton of sense. And so my brother, as it turned out, was graduating from Yale with his public health degree, and calls me up one day and he's like, all right, I'm going to be graduating, but I don't want to be an academic. And I'm like, well, why don't you want to be an academic? He's like, well, every academic I know has a life I would not envy. I'm like, dude, you're talking to me. He's like, it's not about you. I'm talking about me right now. What do you think I should do? And so we sat around talking, and a little bit of background to my brother, Ryan, is he's also computer science, Harvard class of 05. So he was the same class as Mark Zuckerberg. So he actually was in the freshman CS course with Mark. Mark was on his project team. And Ryan eventually at one point blew up at Mark and was like, will you stop it with the Facebook thing? If you don't work on this project, you're not going to graduate. He's like, they already have MySpace. Like, I don't know what the hell you're doing. And in hindsight, he recognizes that was a mistake. And his fraternity at Harvard, not Psi, we don't have a Psi there. His fraternity brother there was Matt Salzberg, and I don't know if people know who Matt Salzberg is, but he approached Ryan shortly after graduation in 2007 and be like, hey, I'm starting a company. And I want you to be employee number one because like you can set up a website like nobody else and and it was Blue Apron, and Ryan was like, well, I know Matt's a good business guy, but like you still have to cook the food. Like how many people are going to order a meal kit kind of thing? And so he turned down that opportunity. And so we were batting around the idea, and we're like, okay, well, if you want to be 23andMe for dogs, like what's the market opportunity for that? And how does that work? Like, is there really a business case there? And batting it around, like from a scientific standpoint, it's really interesting and that's great, but you probably are only going to get kind of like early adopter, like tech savvy kind of pet owner type things. Maybe they already did 23andMe or Ancestry for themselves. And we thought really like, but there's this opportunity if you became like the standard of care, like if a veterinarian thought you needed to have this, right? Like we went from 30 years ago, no dogs had microchips to now 80% of dogs have microchips, right? Like once it becomes like, or if you had like a Carfax for dog model, right? Where nobody's going to adopt a dog until they at least know genetically what they're getting, which makes a lot of sense, right? Like a dog, you're going to spend on average $25,000 over the lifetime of that dog. And your quality of life with that dog depends a lot on that dog's genetics. So like that's one thing you can in theory know before you get the dog, right? When you talk about that, I mean, it's actually a much better business model than 23andMe because there's 4 million people born in the US every year, and there's 8 million dogs born in the US every year. So you actually have twice the market size, at scale. And so now you're talking about a company that can be unicorn billion valuation company if you can solve that. And so after talking that out, we built out a business plan. Investors got excited about it, like even though we were kind of tenderfoots at starting a business. The idea most of the time, like as you test more dogs, your test gets better. Like all of the relatives of your dog have been tested, and you sort of get, and your data allows you to make new discoveries, which makes your test better. And so it's really like this big data play, but you're making money from square one because people are already interested in buying a DNA test for their dog. Right? But like people understand that you can DNA test a dog and learn stuff about it. And so, so it kind of de-risks it a lot compared to other sorts of data plays where you have to like spend a whole bunch of money up front acquiring a bunch of data to get this like monopolizable resource. So, so it was kind of ripe for venture funding, and we've now, gone through Series A couple of years ago, and we're sort of, now trying to figure out where we're going to invest in the future, and whether we need to do another investment round or, whether we're going to go a little bit more slowly just with the revenue that we're getting from kit sales and other partnerships is the open question.
Sure. That actually sort of ties into one of our questions that's been asked. Does Embark have any tie-in to pet insurance carriers?
We we don't have any like official tie-in. I mean, you can definitely see where there is this potential opportunity to, if your dog is genetically healthy, they should get a discount on pet insurance, right? And also, pet insurers, they know a lot about the dog's health. So that could be good for research purposes, right? If you could tie in that information to the genetics of the dog. So it's something we're definitely interested in. It's one of those things, there's so many different things you can do, and how many resources, you have to be strategic in where you put your resources. But it's definitely something that is under consideration.
Cool. Let's see. Another question. Since Embark is an early mover in the industry, do you even have competitors? Are you concerned about them?
Yes. So, obviously every good entrepreneur is going to tell you, well, we're fundamentally different than anything else on the market. We don't really have competitors, but that's obviously, of course we feel that way. We feel like we've created something that didn't exist before, the sort of 0 to 1 kind of phenomenon. But the truth is, if you go on Amazon, there are other companies that are offering dog DNA tests, and they're not offering it on like a research array platform where you can use it to make new discoveries. So we're spending more upfront on the product, but we think it's because when you get a lot more data, first of all, you can at that moment deliver a better product to the customer. But also we value that data because in the future, the discoveries that are going to be able to be made from it. And the proof has been in the pudding. I mean, we've had lots of scientific publications come out. We've got more that are in review that are going to be coming out. So we have actually been able to drive discovery, even though we have not cured cancer yet or anything like that. But that's that's more of the 10-year plan than the 5-year plan.
Sure.
So we have competitors. It was a very real sort of concern. There was originally a patent on doing dog DNA testing that was very broadly written, and that one company had it exclusively, and that company had used that patent to drive other competitors out of business before we came along. But we hired very good lawyers. Sort of the same lawyers that Robert Mueller's firm is from. And they told us, listen, after the Supreme Court rulings for Myriad and Prometheus and all these other decisions, like the patent landscape has changed and those patents are not defendable anymore. And, and so you can always be like, anybody can sue anybody, right? Like you can't stop that, as this presidential election is showing us, anybody can launch a lawsuit. But the fact, they're not going to be able to close you down from this. It's going to get thrown out. You have freedom to operate in the space. And so then it was a matter of, okay, but you're still fighting a competitor that's been doing dog DNA testing for 8 years, right? But they hadn't been innovating. They had sort of, they had a monopoly position, and so they didn't feel like there was a need to innovate at all. And it was a very crappy test. And so, we kind of built something that was a much better user experience. They've been trying to play catch up. It's kind of, it's kind of funny. We wait now to release what our new packaging and marketing is until October, because then they don't have enough time to copy us before Christmas. Right. Right. That's the sincerest form of flattery, I guess. Mm-hmm. And then, there's other competitors in the space as well. Some of them are focused more on dog breeders. Some of them are in other countries. I definitely think competition is healthy. We're not trying to run anybody out of business. We're just trying to make the best test that we can and challenge ourselves to be head and shoulders above everyone else.
Well, since you mentioned it, my dad is a huge dog person. What is the latest I can order a test kit to have it show up under the tree for him?
Yeah, well, this is a weird year for logistics. We've been having some U.S. Postal Service delays, so I don't want to.
Sure.
I don't want to make too many promises. You're definitely fine ordering one now. We've actually had very fast turnaround times for the last 2 months, but before that, there were some postal slowdowns. There was a COVID outbreak at our kitting facility. There was backlogs from COVID at the processing facility, and so we try to deliver results within 3 weeks, and it was getting closer to 6 weeks for a few people, which was rough. But we've done a lot to scale up. It was weird because in March, it looked like everything was going off a cliff. And then by June, it was very clear that, when everybody's staying home, they're all getting dogs and they're all shopping online and they're all buying dog DNA tests. And so it wound up being this, we can't fulfill orders fast enough. Like we are scouring the globe trying to find swabs to put in our kits, right?
Sure.
And in the processing lab, I mean, people are buying laboratory reagents and equipment for COVID testing, which makes sense, but then that puts us in, so it's been an exciting year in the sense that we didn't expect to be profitable, and we're profitable, and sales, have tripled over last year, which is, amazing growth that we hadn't anticipated. And so now it's, can we deliver results and scale up for holiday, which I think we can. I mean, we've hired up now. We've got a new, we now have 2 different kitting facilities with inventory in multiple locations on different coasts and stuff like that. So, we've definitely spread that out.
Cool. Justin asks, you mentioned that the IP reverted back to Cornell. Are they involved in the business?
Yeah, so they're actually, we negotiated a research partnership. So we work with their biobank genotyping samples for them. We collaborate with researchers, not just at Cornell, but other academic institutions, including NIH and some other universities. Because we recognize, I feel like we have a very great core of scientists now. Like we've been able to build a large enough company we can support, a dozen scientists and veterinarians that are doing, research. So we not only have the largest dog genetic database in the world, I think we also have probably the largest nucleus of scientists working on dog genetics. But of course, there are scientists all over the world that have different specialties, in nutrition and oncology and immunology and all that kind of stuff. And so we love entering into collaborations that are just going to get the research moving faster. So there's the Cornell College, logo research partnership on each kit too. We licensed the IP from Cornell, so we had a licensing agreement for that.
Okay. I've got a question from Mickey. How can your research help extend the lifespan of dogs? He says he has labs and 12 years is just not enough.
Right, exactly. That's a perfect question. And I've actually, one of the things Cornell does is whenever a high income individual comes to bring their pet to the Cornell vet school, they take a note of that, and then they send young professors out to talk to them. And so one of the guys I had to talk to was the guy that had golden retrievers, and he was complaining that his golden retrievers, it seemed like the more money he paid for them, the shorter their lifespans were. And, of course, there's lots of things that we can do to improve health and longevity in golden retrievers and Labrador retrievers, and figuring out how to breed away from cancer and hip dysplasia are probably towards the top of the list. And to do that, you need to genetically analyze tens of thousands of Labradors and golden retrievers and figure out who's getting these conditions, and then you can create genetic breeding values, and you can actually breed for healthier dogs that way. And it works. They've been doing it in cattle now for 2 decades for all sorts of different traits. And it works swimmingly, and the genetics will work the same in dogs, but nobody owns 1 million head of dog. So nobody's developed it yet. Mm-hmm. So it's sort of a race to get let's get 1 million dogs in and get this information, and let's do it. But the other thing, which is interesting, is do you know how inbred your dog is? Because that actually plays a big role too. So breeders will tell you, oh, well, line breeding is okay. That's how you fix a trait. And my lines are healthy. And it might be true that inbreeding per se isn't what causes problems. It's the genetic mutations that are getting doubled up when you inbreed. But what we find is across basically every single breed, if you are outbred for your breed, you're going to live 2 or 3 years longer than if you're inbred for your breed.
Okay.
And not only that, like outbred females have bigger litters than inbred females do. Outbred dogs grow up to be larger and more healthy, and they're half as likely to be reported as by the owner to be not in excellent health. So you're getting longer lived, healthier dogs that have better fecundity and better working abilities, just by preferring outbred dogs in your breed, that you're adopting. And you can of course tell the breeder, if you were to cross these 2 dogs, this is the inbreeding coefficient you're going to get. Whereas if you cross these 2 dogs, this is the inbreeding coefficient you could get. So all else being equal, you should do this one over this one, because on average, you're going to wind up with healthier dogs and more dogs. And so we're trying to get we have tools now, we call them matchmakers. So a Labrador breeder can test the dogs that they're interested in breeding and compare them to other Labradors. And then, okay, well, these 2 I don't want to breed because they carry the same genetic mutation. So I don't want to, but these 2 dogs I could breed, but it'd be a higher inbreeding coefficient in the litter than these 2 dogs. And so you can make those decisions.
Is that the type of thing that breeders list? Like if they've consulted with you or tested their lines?
Yeah, I mean, the ones that are kind of on the cutting edge, yes. I mean, we're still at the stage where, most, at least many breeders are still uncertain about genetic testing. Mm-hmm. It's still in its infancy. They've used pedigrees, and pedigrees, of course, give you inbreeding information, but it's not nearly as accurate because they only go back 5 or 10 generations. Mm-hmm. And the inbreeding in labs, a lot of it's coming from even further back. And it depends not just on what the family tree looks like, but it actually depends specifically on which parts of which chromosomes came from which individuals, sure, which you don't get from a pedigree. You can only get that by looking at the DNA. And so in fact, in a pedigree, all of the litter mates are going to have the same estimated inbreeding coefficient. But in fact, when you test them, you'll see that, oh, well, this dog is completely inbred on chromosome 2 and halfway inbred on chromosome 21. And this one has a little bit on chromosome 3, but it's mostly outbred. Like you actually get different inbreeding values when you actually go like gene by gene looking across the genome.
Sure. Now, if I sent you a test from like my dad's dog or something, would you be able to, would the report that I got back tell me like he might have a propensity for heart disease, so you should switch him to heart healthy food? Is it as granular and specific?
No, so that's what we're trying to build. So the report will tell you whether or not your dog is at risk for exercise-induced collapse, right? That's a single gene disorder. We can test the mutation for it, and your dog is either at risk or it's a carrier or it's clear. We can tell you whether your Labrador has the gene for increased appetite. So there's a Labrador specific mutation that like over 10% of Labradors have, and it gives them insatiable appetites. And it's really only found in retrievers, specifically labs. So it's a very recent mutation, and it's increased in frequency, not because people want fat labs, but it's because Labradors that are insatiable are really easily trained. Huh. And so we can tell you whether your lab has that. And we can tell you about all the coat color, genes that it carries that we know about. And we've got a new coat color paper that's going to be coming out shortly. But what we can't do is these complicated genetic things. Like your dog has a 2-fold increased risk of lymphoma, but a 50% reduced risk of, this kind of heart disease, mitral valve disease or tricuspid valve dysplasia or something like that. That's what we're trying to build up the data set to do, and now we're getting close to 10,000 labs in our database. And so, the more of those labs that get older and we get veterinary records from, the better we're going to be able to train. I mean, we could train something now with the data that we have, but it won't be very accurate.
Right.
So I don't want to put something out that's not like scientifically validated to a certain level of accuracy, because people do make choices about how to, raise their dog, what to give their dog, which dogs to breed. You know, all these have downstream consequences. So you want to make sure, you know, that your tests are accurate before you offer them.
Sure. Mark asks, has your experience with Embark sparked other entrepreneurial ideas that you're running with?
I think I've got my plate full right now. It is neat kind of going through the process of building a company from nothing, and then you can sort of see which kind of, sorry about that, which kind of opportunities are one, you know, like you could look at stuff and be like, there's a better way of doing this, but there's not really a good business plan for how to get there, or it's not a big enough market to make it worthwhile. But then, you know, yeah, as you go through, you can see other areas where it's like, yeah, there's actually an opportunity here. If I had more time on my hands, or if I had the right, you know, maybe it's CTO, or maybe it's COO or something like that, you could probably get something off the ground. Yeah, I mean, and it's sort of like in research too. You're always looking for, well, this has been studied, but you know, like you've got this interface here where we really don't know what the answers are, and it's a tractable question now because this tool got developed, and you know, like, so it's all of that kind of stuff. It's, you know, a little bit thinking a few steps ahead about what is going to be in demand in a year or two, and what can I do to address that.
Sure. To switch back to Cornell for a minute, now that you're back from sabbatical, what are you studying or researching, or are you mostly teaching?
Right now, I'm mostly teaching, and then it's going to be grant writing to try to build back up the lab. So it's more right now just kind of bioinformatics support for some existing projects that, you know, have been going on, where you know, because the vet school is full of veterinarians that have really great insights on disorders and can get samples, you know, from their clinics, and just kind of need help going through and analyzing and figuring that out.
Okay.
We were doing some conservation work earlier. I actually had a student that just graduated with her PhD, and so she worked a little bit with dogs, of course, but her main work was African painted dogs. So I was jealous because she would get to do field work. And then, you know, of course, the same sorts of inbreeding issues and, you know, and whatnot that we were doing with dogs, she could do with African painted dogs.
Very cool. Let's see. One question we always like to ask, have you run across other Psi-Us in your professional life?
Well, I see Tom like a little less often now, because the Cornell chapter is suspended for a little bit. So yeah, Tom Fox, he's always like that uncle you see at funerals, because it was like every time he was coming over, it was like because the Kai had done something wrong. So it was like, hey Tom, it's great to see you. I wish it could have been under better circumstances. So got into travel around. You know, when I was at Stanford, Bob Pekelnicki came down. We got to have you know, dinner together, which was great. In 2017, Embark sponsored Puppy Bowl. We actually genetically tested all 72 puppy participants at Puppy Bowl. And if you are faint of heart, close your ears now. But Puppy Bowl is a lie. Those dogs have all been adopted out well before the broadcast. It's taped over the course of 2 days in October in New York City. But now that I've said that, they are all shelter dogs. And they do events down at the Super Bowl too. So I got to go down to the Super Bowl, not to the game itself, but to a side arena with puppies. But we were down there in Houston during the Pats' big comeback against the Vikings. And John Drew actually came up, so he got to see Embark Center there for Puppy Bowl. So that was fun to get to hang out with him. Yeah, I've crossed paths with a few Psi-Us, you know, from the chapter over the years, for sure.
Excellent. One of the things I'm looking at putting together is trying to connect all of the Psi-Us that are in the sciences, biologists and geneticists and PhDs and such. So I might be in touch with you about that to see if you want to meet some other
Yeah, for sure.
biologists sometime in the coming year.
Yeah, no, there's a lot of excitement in the field. There's a lot of cool new stuff, coming out that wasn't even on people's radar 10 years ago, in terms of epigenetics and ancient DNA and all sorts of stuff.
Very cool. Have one more question from Cameron, and then I'm going to do a little more housekeeping, and we'll come back and pick up any questions we have left. Greetings, Dr. Boyko. Great to see you. Curious to hear your take on the implications of intentional breeding at scale as breeders choose favorable traits over unfavorable ones. Will it alter dog population for better or worse? Or perhaps it's such a small percentage of total breeding that it is inconsequential.
Yeah, so each, for a lot of these traits, like let's say Dalmatian spots, right? Mm-hmm. Like that's one part of the genome. So the rest of the genome, like if you're trying to fix a breed like Dalmatians to have these big spots, you're not really losing too much diversity, as long as you're not only breeding like 3 dogs, right? Like as long as you're just like enriching for those spotted dogs, there's enough time to retain the genetic diversity on all the other chromosomes. But that one region of chromosome 3 that has the spot modifier locus, what happens is it becomes fixed, and not just that one mutation, but everything, within, could be 1 million base pairs away, right? Like everybody's got the same genetic code there. And so one problem that happens when you do this kind of like strong selection for something like that, is that if there's anything wrong with the genetic code, you're accidentally selecting for it when you're selecting these spots right there, and it become fixed. And that actually happened in Dalmatians. So right next to the spotting locus, there's a mutation in a gene that causes bladder stones in Dalmatians. And so it's completely fixed. There's nothing you can do about it unless you CRISPR a new Dalmatian, and the FDA doesn't want you to do that, so you can't do it. And so you just have to put them on special diets and stuff. So that's one thing that happens. But yeah, the other thing is these are closed populations. So once there's no way to bring diversity in without reopening the stud book, which is something that 99% of breeds don't want to do. Or maybe 100% of breeds don't want to do it, but 1% of them kind of recognize that we're so far gone, we have to do it. And so you get this inexorable kind of loss of genetic diversity if your breeding population is too small. And that is certainly the case in many, many dog breeds. And that population becomes smaller if you're only choosing to breed dogs that have a very certain trait. And then I guess the third thing is your power for selection is greatly lessened if you're trying to select for many, many things at once. Mm. And so if you're trying to select for a particular conformation, then your ability to select for health is going to be reduced. Like you need to focus, or you need to create a balance, right? But so the more that you are concerned about fixing a certain trait, then the less power you have to also select for health and longevity. So it's sort of a triple whammy if you're trying to do too much. And so you want to, yes, you want to breed towards whatever standard you want, but you want to do it in a responsible way. And that responsible way isn't reducing the gene pool to just 1% of the dogs, because in almost every breed, that's going to be an unsustainable loss of genetic diversity over time.
Okay. All right. Thank you, everybody, for coming to listen to us this Thursday night with the virtual speaker series. We've got a couple upcoming events and announcements. Those of you that are undergrads or grad students or advisors to undergrads or no grad students, scholarships are now open on psiu.org. So the applications for our need and merit-based scholarships and our new grad student scholarships are available now at psiu.org. Coming up next week as part of the speaker series, we have the 2020 Economic Symposium Look Ahead. We'll be speaking with 5 different industry-leading professionals who are PSIUs in the economic sphere, investment, macro and micro, and we'll be sort of trying to gauge what the next 6 months will hold for the economy and for investment. And then we have a speaker series year-end review in December. Adam, you of course will be invited to participate if you wish. We'll be showing sort of a highlight reel from different speakers this year, looking towards some of the speakers that we've arranged for next year, and just sort of catching up. And that'll be more more casual, and people can catch up on what they've been doing. As next steps for our alumni who are listening, if you have not, may I suggest that you follow along upcoming events at psiu.org. Register for upcoming speaker series as they strike your fancy. They are archived on our YouTube channel, which you can reach through psiu.org. And the Psi Upsilon Professional Network on LinkedIn is somewhere that I encourage all of you to share your research papers or white papers, or post needs for jobs or interns, or look if you yourself are looking. It's an excellent resource on LinkedIn. And of course, I would be remiss if I did not say that donations to the foundation, which is a 501c3, are tax deductible, and any donation helps support our programs, operations, publications, and outreach efforts like the speaker series, which I do hope you have all enjoyed this evening and over the year. With that, I'm going to stop share. We're going to go back to Adam for just a sec. It does look like we've had a question or two pop up.
Man, I'm falling behind.
Yeah,
I don't have any good DNA from Clyde. That is my one regret. And I think eventually the house probably got cleaned up well enough. We don't have any DNA left from him there either. Yeah.
All right, so for those who were not undergrads with you, who was Clyde?
So Clyde was a shelter dog. Flounder and I adopted. So he was a shelter dog that we adopted, and that was Clara, my daughter. And he kind of had his run of the house, I would say, and he had this amazing ability to escape from the house and then invariably be found at a sorority house. Like we would get a call, and then like we would show up, and it would be like some girl, and like Clyde's in her arms, sort of thing. It's, have I done well? Yes, you've done well, Clyde.
All right. Any particular breed, or just sort of a brown dog?
He looked like a lab hound mix, but honestly, I'm only good at figuring out the breed if I have the DNA.
Fair enough.
Looks can definitely be deceiving.
All right. Well, with that, thank you, Adam. It has been an absolute pleasure.
Thanks, Jonathan. Yeah, I've really enjoyed it.
Great. Yeah. So we'll have the end of year roundup, and I'll touch base with you afterwards.
Sounds good. Sounds like fun.
Have a great evening, and talk to you later.
Thank you. Thanks.
Thanks everybody for series. As always, you can reach me through psiu.org, and good evening.